Right, I think it's time to kick off. So thank you so much for joining the webinar this morning on the Animal Free Antibodies database. I'm Dr. Stephanie Modi from Animal Free Research UK, Science Manager there. So let's have a look at what we're going to look at today. So here's an overview for you. I'm going to start with an overview of the antibodies landscape. So talking about antibodies as biomedical tools, the antibody market, and we've all got different levels of knowledge on antibody production. So I'll be going over how antibodies are made using animals and the animal-free and recombinant alternatives that are out there. Also talking about the benefits of animal-free antibodies and recombinants, and in light of these benefits, why researchers are still using animal-derived antibodies. I'll then go on to talk about how the database works that we've been developing and the next steps in driving the uptake of animal-free antibodies. And hopefully there will be time for questions at the end. So this is a very quick snapshot of antibodies as tools. It's non-exhaustive, but here are some examples. In blue, you can see that they're immensely useful as tools in research for detecting targets of interest. So, for example, in ELISA or Western blot, etc. And our database is very much focused on that audience, on academia and research. But antibodies are used really widely across the board as therapeutics, for example, to treat cancer, autoimmune disease and rheumatoid arthritis, and also in tests, so to test for fertility and pregnancy, substances of abuse and performance-enhancing drugs, and also to detect disease, so for example, sexually transmitted diseases, or we're all familiar with COVID tests where antibodies are used in there. Also for safety, so quality control, predicting the safe limit of ingredients in cosmetics or household products, and also to detect allergens, hormones, metals, and pesticides in our food and drink. So that's just to show you that antibodies are really widely used across a whole range of applications, which leads on to this slide, which is the antibodies market. The antibody business is a massive business. And just looking at the shape of that graph, you can see that the antibody market is growing rapidly. In 2023, it was valued at $162 billion, and it's rapidly estimated to grow to over $277 billion by the end of 2028. So big business, and it's growing. And as I mentioned, our database is focused at very much the research-only antibody market, and that itself generates between $2.2 billion and $2.7 billion each year, with more than 300 companies producing these antibodies. The largest market share belongs to North America, but the Asia-Pacific market is expected to grow at the fastest rate. Some of the reasons behind that are the improvements in healthcare infrastructure, increased amount of chronic diseases, and also some of the laws governing antibody production are a bit less stringent in those countries, which does have an impact on animals, which I'll talk about a little bit later. And the European market is also expected to grow significantly over this time. So it's a big business. And why is it growing? Well, we have about 20 to 25,000 genes, and each of these genes codes for a protein. And each of these proteins could potentially represent an antibody target. But the number of antibodies that we're actually producing, or that the suppliers are producing, could actually exceed this tenfold. And that's because antibodies are being produced that can identify really closely related targets. So it might be that you have an antibody against a particular protein and then a phosphorylated version of that protein, for example. Or there can be targets with really subtle changes through alternative splicing or post-translational modification. So with these huge numbers of antibodies that are being produced, obviously this is going to impact animals that are used to produce these antibodies. it will mean greater suffering for animals endured, and really highlights the need for us to look for alternatives to replace animals used in antibody production. So the next couple of slides is a reminder on how antibodies are made using animals, because we all have sort of different levels of knowledge on this. So first of all, polyclonal antibody production. This is one of the oldest ways of producing antibodies using animals, and it actually represents the biggest market share of antibodies at the moment. Polyclonals represent 60% of the market share. So a huge range of animals are used for this, as you can see. So from mouse to rabbits, llamas, chickens, pigs, goats, camels, huge range of animals. And for the antibody that you want to produce, the antigen is injected into the animal, for example, a mouse, as you can see. And the animal is usually repeatedly injected with the target antigen, which kicks off the immune response, which means the antibody of interest can be generated. Ultimately, the animal is bled and the antibody is purified. And then you end up with a batch of polyclonal antibodies, poly meaning many, which means that these antibodies can identify different epitopes on the same antigen. So you get a mixed bag there. There are some issues with using polyclonal antibodies. So for example, it can take a long time to make them. You've got to consider all the animal costs, housing the animals, training people in animal handling and immunization techniques. And they're not sequenced. So there's no long-term reproducibility for these antibodies. And they often hit targets which are not intended targets. And you get batch-to-batch variation. So this is going to really impact your results if you're getting a different set of antibodies each time you immunize a batch of animals. And you may get unwanted contaminants as well. So this method of antibody production was joined by another method called monoclonal antibody production. And this was developed in 1975 in order to generate antibodies that were more specific for targets mainly. So again, the animal is immunized with the antigen to generate the antibody of interest. But this time, the monoclonal antibodies are generated by harvesting B cells from the animal's spleens after the repeated immunization. And these B cells, which are the antibody-producing cells, will be fused with myeloma cells, which cancer cells. So they have that feature of cancer, which is immortality. So this fusion creates cells called hybridomas. And these hybridomas have features of both the B cells and the plasma and the myeloma cells. So they're producing your antibody, but they can also grow in an immortal way. And once you've got your hybridoma, there's two main ways that you can culture and produce the antibodies. So you get your monoclonal antibody, so mono meaning one, which means it's specific for a particular epitope on an antigen. And the two ways are, firstly, ascites. This is whereby the hybridoma is injected into the abdominal cavity of an animal, into the ascites fluid, where it can multiply while the fluid is generated, and then you can harvest the monoclonal antibody multiple times. There's a lot of ethical issues around this method of in vivo antibody production, because the animals experience a lot of pain and distress and suffering through this method. And it's in fact banned in a number of countries, Canada, New Zealand, for example. But you don't have to produce antibodies in this way, monoclonals. You can do it in vitro. So what you can do is, in fact, culture the monoclonal antibodies in vitro in a dish in animal-free serum. And then although the initial generation of the antibody was using an animal, the antibody can then grow indefinitely in vitro, in addition, animal-free media. There are some issues with monoclonals. So the idea is you get a single clone, but in fact, hybridomas have been known to produce more than one antibodies. Also, again, they're not sequenced, so you don't know the sequence, it's not reproducible. And in fact, these monoclonal antibodies are not always stable. They can suffer from genetic drift over time. So the monoclonal that you've been culturing all this time will suddenly change, and that will affect your experiments. And also cell lines can die as well. So issues with those as well. And what about the numbers of animals used to generate these antibodies? So ECVAM, which is the European Centre for the Validation of Alternative Methods, estimate that around 1 million animals are killed each year in Europe for antibody production. And it's somewhere between 5 and 13 million animals globally. The number actually depends on the species used, the amount of antibody required, and the success of the immunization. And the true magnitude is actually unknown because a lot of countries don't publish statistics on the number of animals used for antibody production. To give you a rough guide, in terms of the production of polyclonal antibodies, around 2 rabbits are used, or 5 to 10 mice for just one batch. And for monoclonal antibody production, typically 5 mice are immunized per hybridoma protocol. So a big impact on animals. But what about accelerating the adoption of animal-free antibody alternatives? So there's quite a well-known directive that came out of the EU, called the 2010-63 directive, that said, wherever possible, a scientifically satisfactory method or testing strategy not entailing the use of live animals shall be used instead of a procedure. And then in 2020, the European Union Reference Lab for Alternatives to Animal Testing, called EURL ECVAM, and this is a division of the Joint Research Centre, which is the Science and Knowledge Service of the EC. They came out with some recommendations in 2020 for the phase out of animal-derived antibodies, stating that in purple, as you can see, EU countries should no longer authorise the development and production of antibodies through animal immunisation, where robust, legitimate scientific justification is lacking. So at the moment, momentum is building for the scientific community to speed up adoption of these non-animal methods, especially in light of their benefits, which I'll go on to highlight in a later slide. So what about non-animal derived alternatives? Many of us are familiar with recombinant antibodies. So these are antibody fragments that are produced using antibody coding genes, and they usually comprise a heavy and light chain of the variable region of the immunoglobulin. In terms of antibody production, there are different stages to consider in the production of antibody. So there's the discovery of the original antibody clone. There's also the actual antibody production stage and the final formulation of the antibody. In terms of the discovery stage, the actual gene source can be completely non-animal derived. It can be a human source or synthetic, or the discovery can be an animal source. So it can be from your hybridoma cell line or from your B cell. Thereafter, you can clone that gene, and then you can use in vitro affinity selection using display methods like phage display, ribosomal yeast display. So the phage is able to display that antibody on its surface. And then you can use clonal selection and sequencing to get your antibody of interest and know its precise sequence. You can then transfect cells, and you can produce a cell culture that produces your desired recombinant antibody, which you can then collect and purify. Important to note, and something sometimes overlooked, is that in this stage, or the various stages of antibody production for recombinants as well, animal-derived biomaterials can be used. Say, for example, fetal calf serum, and there's a lot of ethical issues around the use of fetal calf serum, and also bovine serum albumin, often used as a stabilizer, for example, in final formulations. But these can be replaced by animal-free alternatives. Big advantage of your recombinants is that they're sequence defined and they're reproducible, meaning that your results are going to be more reliable and reproducible. So I've put this slide in to show you an example of an actual supplier in terms of how they produce their antibodies using plants. So this is Cape Biologics, who are based in South Africa. And what they do is they get a digital sequence of DNA, which is a bit like a computer code. In terms of the source of their sequence, they would get this mostly from published sequences out there in the literature or on databases, for example. So it might be that in order to get this sequence, it could have been an animal-derived sequence 50 years ago, where the animal was immunized 50 years ago. It could be historic immunization, or it could be a non-animal derived sequence. So what they do is they synthesize this computer code into physical DNA. Then they pop it into a vector, which is like a carrier. And then they pop that vector into a bacteria. And we know that bacteria, they have the feature of being able to multiply and multiply and multiply. So what they can do is then deliver this bacteria into the plant cells, and the vector actually hijacks the plant's protein-making machinery. So essentially, the plant becomes like a mini bioreactor. Then you can harvest the leaves of that plant, and you can purify and quality test your antibody. So this is how one supplier makes their antibodies from plants, as an example. Here's another example of a supplier who manufactures using mixed methods. So this is GeneTex in America. So for their discovery phase, they use animal immunization to create the initial library. So as opposed to Cape Biologics, who use historic immunization, GeneTex use rolling immunization. So they're doing this with new animal involvement, so it's a little bit different. For their production phase, their downstream production phase is completely recombinant, so they don't use animals. But they do use animal-derived biomaterials. They use fetal bovine serum for their cell culture. And then in their final formulations, they also use bovine serum albumin as a stabilizer. So take home here is that a lot of people think about recombinants as animal-free, but actually, if an antibody is recombinant, it doesn't mean it's completely an animal-free antibody. It could be that animal immunization was used in the discovery phase, for example, or the biomaterials are being used. So I've put this reference in simply because it's a really good overview. It's in Nature Biotechnology by Alison Gray of the different methods used to produce antibodies and the issues around them. So if you're not that familiar with antibody production, it's a really good article to read, Animal-Free Alternatives and the Antibody Iceberg. So she has some diagrams in there which basically summarize the production methods that I've talked you through. So polyclonal, monoclonal, recombinant, and the issues around there. So I recommend that to read. And she also has this picture of an iceberg, which basically shows you that recombinants and animal-derived antibodies are merely the tip of the iceberg. And there's so much development and new technologies going on under the surface that are up and coming as well. I'm not going to go into these in detail, but I will mention some of them that are on the horizon being developed at the moment. So we've got things like molecularly imprinted polymers, which are made using computer-assisted modeling. Affimers, which are small engineered binding proteins, aptamers, which are short single-stranded DNA or RNA molecules that can bind different targets, DARPins, which are genetically engineered antibody mimics, monobodies, which are synthetic binding proteins, and anticallins, which are artificial proteins that can bind to antigens. So there's a huge range of animal-free replacements that are being developed currently. So I mentioned earlier that there's lots of benefits around animal-free antibodies. I want to highlight some of these benefits. So if we look at the big orange circle there, you're going to get superior batch-to-batch variation. You're not going to get that problem with having to inject a different set of animals and then get a different set of antibodies. And this is going to avoid misleading data. It's also going to save time and money in the long run. And also recombinants hit their targets more accurately. So a 2008 study showed that fewer than half of around 6,000 routinely used commercial antibodies recognized only their specified targets. Sustainability. So if you have your sequence-defined reproducible recombinant antibody, you're going to be able to get a renewable and unlimited supply of antibody. And it's not just limited to what you can get from that animal in its lifetime. We've mentioned the huge numbers of animals that are used in antibody production. So it's a more ethical option. And it's also more effective, cost effective. You're not going to have to pay for animal housing and upkeep, staff training in handling the animals and the immunization technique. And if you've got that reproducible stock of sequence-defined antibodies that you can keep producing, although there may be some cost to set up the library in the first place, in the long run, it's going to be much more cost-effective. They're also faster to produce. So monoclonal antibodies take between 4 and 15 months, and recombinants can take between 2 and 4 weeks to produce. So there's in fact a staggering loss of $800 million estimated to the biomedical research community from using animal-derived antibodies. So if there's all these benefits, why are people still using animal-derived antibodies? So one of the reasons is that if scientists are used to using a particular antibody that is working for what they want it to do, there's an inertia and a tendency to use what you're used to. So there's some initiatives that are going on at the moment, for example, where like the recombinant antibody challenge, for example, whereby researchers are being encouraged to try a recombinant antibody alongside their polyclonal, for example, so they can compare and contrast and be assured that the recombinant will work just as well as what they're using. So there's also a lack of awareness of the advanced level that's now attained by non-animal derived antibody technology. So it might be that when this technology came out in the 90s, there was a bit of bias against it. It was viewed as unreliable and expensive, and there's a bit of a hangover from that still. And these sorts of misconceptions affect suppliers as well. Because, for example, with small companies who are producing antibodies and might have slim profit margins, they're then somewhat dissuaded in investing in this technology. And ultimately, that will lead to a shortage of companies who are producing the antibodies. And up to now, there's been very much a focus on the more lucrative therapeutics market by antibody producers. And from a user point of view, it's hard for people to know where to find the animal-free alternatives as well. Hence the database. So this database that we're developing is really vital in overcoming some of these obstacles through increased awareness of the benefits of animal-free antibodies, more education for users, helping people to find and access the antibodies, and also demonstrating to suppliers that user demand is there for recombinant and animal-free antibodies. So now I'm going to go on to talk more specifically about the animal-free antibodies database. It's a database that's completely free for users and suppliers, and it offers well-defined information on the methods used by suppliers in all the stages of antibody production. So that's discovery, production, and formulation. And the entirely animal-free antibodies are prioritized in the search as the green antibodies. But we also offer a redirect to manufacturers who can custom make animal-free antibodies if a suitable animal-free antibody can't be sourced. And we mentioned that education is really important. So there's a host of information on the scientific, ethical, and financial advantages of using animal-free antibodies. And researchers can link to our community of practice and join forums where they can share information, expertise, and experiences on using these antibodies. And really important for suppliers as well. They'll be able to track demand for their antibodies and for animal-free antibodies. And hopefully this will instill motivation or market pressure within the biotechnology sector to encourage people to shift towards animal-free antibodies. In terms of where we're at with the database, the development is complete, and I will show you some screenshots of what we have. We will be moving live shortly, whereby we will be onboarding suppliers to get the site really well populated. We currently are on a staging site, and we have 22,000 antibodies from just 4 suppliers. So the next stage will be onboarding more suppliers, and we will announce the launch more widely to users soon. So here's a few screenshots, sneak preview of what it will look like. So this is your home screen where you've got your search box for your antibodies, and you can see a number of tabs at the top. So the database is housed within the website. You've got your About Us tab, the forum that I mentioned, benefits of using animal-free antibodies. There's a link to our community of practice and the forum, and some examples of all the benefits of recombinant antibodies, and also some references. So it's evidence-based, and people can find reliable references to back up these advantages. So in terms of the actual database function, there are a number of ways that you can filter to find your antibody of interest. So I'll come back to antibody origins in a minute. You can filter by application, Western blot, ELISA, etc. You can filter by antibody type, species reactivity, or supplier. But I want to show you more in detail about the antibody origins, because that's a really key part of it in terms of offering that really well-defined, clear information on how that antibody is actually made at each of those stages, at the discovery stage, at the production stage, and the use of animal-derived biomaterials. So this is what it will look like. You'll be able to tick different options for what you're looking for. So for example, in this filter, animal-free discovery, animal-free production, and no use of animal-derived biomaterials has been ticked. So that will be your absolutely animal-free green antibody. And what you'll get is a little box telling you what you've selected. So you've selected category A, which is the animal-free discovery and production with no animal-derived components used, and your antibodies are recombinant and sequenced. And then you'll have your antibodies there, which you can click on. You'll be redirected to the supplier site where you can check the prices and purchase if you wish. As you can see, there's little question marks next to each filter, and that's to help guide the user on what these actually mean, because not everybody is that familiar with the ins and outs and details of antibody production. So for example, with animal-derived discovery pre-2020, this little box, if you click on the question mark, will explain the reasons behind that. So this is an antibody that the original claim was discovered pre-2020, that date chosen because it's the date of the ECVAM recommendation that came out. So it might be that immunization of the animal is historic and happened a really long time ago, and there's no new immunization used by that supplier. So that would be in contrast to someone like GeneTex, who would be using discovery post-2020, and they're using rolling immunization and new animal use. So it's essentially to distinguish between historic and ongoing animal use. This is the little question mark I selected for FCS, and it gives you a little bit of background on what FCS actually is, if it's something you're not familiar with, fetal calf serum. And this is animal-free discovery, tells you a little bit about what that phrase actually means in terms of sourcing the actual genes. So yeah, this is very much an education piece as well, because we recognise that more education is needed for both users and suppliers, and users need guidance on how to interpret the information. This is another example. So different filters have been selected, and again, you get a little category popping up. So you have selected animal-derived discovery, immunization, animal use in discovery occurred before 2020, so that's historic immunization with no new animal involvement. Production's animal-free, so it's recombinant, but they have used animal-derived biomaterials in this case, and the antibodies are recombinant and sequenced. So again, this offers the user transparency on the production and free choice in terms of what you're willing to accept as a user in terms of animal use. But obviously a big step away from things like polyclonals and ascites, which are not accepted on this database. All the antibodies are recombinant. So a few points about this database, which is why it's so unique. As I mentioned, it offers well-defined, transparent categories showing discovery and production. And as mentioned before, some recombinants are stating that they're animal-free, but actually, from what I've shown you, you can see that the discovery phase may involve animal immunization, whether it's historic or ongoing, and there may be animal-derived biomaterials used, and users want to know this information. So the database will add this transparency and also potentially a new pressure on suppliers to move to less animal use in their discovery methods. The categorizations might also be really useful for antibody developers, and we have, in fact, 10 categories. And it will be useful for developers, users, project license authorizers, and funding bodies. So, for example, a funder might only accept applications using antibodies for category A, for example, or an end user in their publication might say, I only used antibodies in category B or A for this publication, this research. And it might potentially be useful for the granting of licenses as well, for in terms of what the Home Office license inspectors do. And it will also overcome difficulties for users. A lot of the feedback has been that people really find it hard to find animal-free antibodies and understand how those antibodies are made. So this overcomes that obstacle. And it's very much an inclusive database. It's not just the 100% green animal-free antibodies. It really includes, and as I've shown you, lots of suppliers using mixed methods. And that means that suppliers will be able to track demand for their antibodies. And hopefully, if they see that there is this user demand for the animal-free antibodies, it will encourage them to transition to more ethical categories over time. Yeah, so that's pretty much saying the same there. But don't take it from me. Here are some expert opinions. So João Barroso, scientific officer at the EU, said what's needed is well-defined categories which are well understood by users and suppliers. so users know what they're buying. Susanna Lohimier is at the EC. The most common question is, where can we find animal-free antibodies? Nick Turner is working on molecularly imprinted polymers, and he said that being able to have a pathway for suppliers is really important to transition up those categories. Alison Gray, she said manufacturers need time to develop. They can't switch overnight. So this database is very much something which the change will happen over time, hopefully led through user demand. Esther Wenzel, who makes completely animal-free antibodies using human sources at Upcalis, said the database will add pressure to higher standards in the discovery phase and not just production. We can't handle all the requests we get for animal-free antibodies. The market is huge. And this is something when I spoke to some antibody suppliers, they weren't aware of this user demand. We've mentioned that education is really important. So Michael Fiebig said part of doing this job is to educate people. Probably about 10 to 15% of researchers know about antibody production. And in a couple of years, it could be that every PhD student knows about this. So we want to educate and make people more aware as well. And Jeff Pilkington, who's Professor of Neuro-Oncology, has talked about the market forces. The ability and willingness of scientists to use the products and biotech and commercial companies to go into the market is based on that demand. It's all about transition. So again, it's that user demand, which is going to add that pressure to suppliers to meet that user demand. So the final slide is just talking about the end goal that we are heading for. How will this database ultimately help to phase out antibodies made using animals? So the current state is that non-animal derived antibodies aren't as widely available or as affordable as animal derived antibodies. Although the prices do vary, a lot of them are a very competitive price. But a true shift to animal-free antibodies is really only achievable when end users demand them and when they become the default choice of academic and commercial labs across the world. So what we're doing through this database is empowering users to make well-informed choices through those clear categories and giving them well-defined information on the origin. And this will help to drive up the demand for animal-free antibodies. So if improved quality, that scientific quality from recombinants and ethical standards are demanded from the scientific community, then manufacturers will be under more pressure to invest the time and money to transition to meet the user demand. So we really need the scientific community to place this pressure on, and scientific community stands to benefit through reproducible and renewable sources of antibodies which have greater versatility and also meet a higher ethical standard. So that leads us to the end goal, which is that non-animal derived antibodies will be as widely available and affordable as animal derived antibodies are today. And ultimately, we want these animal-free antibodies to become the more advanced, ethical and more scientifically excellent new standard. So thank you very much for your attention on this webinar. I hope that you found it really useful. I will stop sharing my screen and I think we have a few minutes for questions, so I can take any questions that anybody has. Okay, I'll just stop sharing if I can figure out how to do that. All right. There we go. I think I've stopped sharing now. So I will, you can put up your hands or there's a few questions in the chat. Let's have a look. Is there a chance to receive a certificate of attendance? I'll look into that for you and get back to you, Elka. This is outstanding, thank you. You mentioned the possibility of users adding reviews. Can you talk a little bit more about that functionality? Yeah, that's a really interesting point. So on the homepage, we have a place for testimonials, and we're hoping to populate that more with testimonials from people who've actually used antibodies, animal-free antibodies or recombinant antibodies, so people can see that really easily. And there's also the community of practice that I mentioned. So we have our forum where people can share and exchange information on that as well. Excellent presentation, such a lot of effort and work, well done. Thank you so much, Linda, really appreciate that. Angela, also, can you provide any more details about your planned timeline, launching in a few weeks or months? All I can tell you at the moment is that the development is complete. It's been user tested, it all works fantastically. We are onboarding suppliers, as I mentioned. We've got 22,000 antibodies on there and our 4 suppliers who have helped with the development. And the next stage is to onboard more suppliers to make sure it's fully developed. So going live, it will be imminent and people will be able to find it, but the bigger marketing plan around the launch will be later. Not able to give you an exact date, but we will keep everyone posted on that. It's not live now, no, Alison, but it will be live very soon so that we can onboard suppliers to the live site rather than the staging site. Catherine, thank you for your presentation. You mentioned non-animal antibody alternatives. What are your thoughts on increased uptake of these in research on a larger scale? We're actually hoping possibly more in a phase 2 part of the database development is to include these sorts of antibodies in the database as well, because I think for me this is very much the future of antibodies. You know, if you can get those sequence defined really specific molecules that are going to give you your reliable reproducible results, which is the key driver for scientists. I think these are going to play a massive role in the future, and so do people who are working on them, like Nick Turner, who I mentioned. Alison, one barrier we had from Plantify is the availability of sequence data. Can you comment? Yeah, that's an interesting one. I know that there's a lot of intellectual property issues around sequence data, because obviously if a company immunizes an animal, for example, and then has the sequence for that particular antibody, they're not going to want to share it and make it public. So that is a barrier at the moment. I'm not sure how that will be overcome in the future, but I know that there are a lot of people who are sharing on different databases. For example, there's the ABCD antibodies database, which is aiming to make the sequence data widely accessible, and also different publications. But that is a barrier, and that's something that needs to be overcome, because if we're not sharing the data, the sequences, then we're going to have to, different suppliers are going to have to keep immunizing when they wouldn't have to if that sequence data was public. Producing antibodies in plants, technically modified. Will we need an S1, S2 lab for using the antibodies afterwards? I'm not sure. I'm not that familiar with what you mean by S1, S2 labs. Elka, are you willing to put your mic on and elaborate on that for us? Or you can type it in the chat. We're coming up to 10:45. So is there anybody else who has a question and wants to put up their hands and ask a question or pop anything into the chat? But obviously you can get in touch with me at Animal Free Research UK if you have any follow-up questions on this, and we'll certainly be keeping in touch with you in terms of the development of the database. So final one from Elka then. So using genetically modified organisms, safety lab is needed. In terms of, so you're thinking about what Cape Biologics are doing. So I'm not sure specifically what sort of labs they are using, but I imagine they are using the appropriate safety labs. I don't know too much about that, but that's something that can certainly look into. So we're at 10:45. So just want to thank you all for your attention, and I hope that you found that useful and informative, and wishing you a very lovely rest of the day. Thank you, everybody.